Samantha Ferrao. She's a biotech undergrad student who went on to pursue her master's at Ohio State University, and she's now doing her PhD in biomedical engineering at Virginia Tech. Today, Samantha will share with us her journey from biotech to biomedical engineering, key lessons from grad school and her research, latest trends in the biomedical field, and the future of medicine through nanotech. So, Samantha, the mic is all yours. You can go ahead.
Okay, so hello everyone. Hope everyone's had a good day and is having a good evening. So, as Anushka said, I'll be presenting on my career journey, the lessons learned throughout grad school, and what to currently expect from the biotechnology / biomedical engineering field. And yeah, so, what do I expect from my talk? So, I'll briefly give you all an introduction about myself. Then, I will venture into my journey so far. And then, after which, I will pivot into the advancements in cancer and tissue engineering, which is my main realm of research. And then, the current biotech trends and market, which I will briefly touch upon. So, firstly, about me. So I started out in undergrad at BITS Pilani Dubai campus. I was from the batch of 2016 to 2020. And as you can see here, I did my undergrad in biotechnology. And then after which, like the pandemic happened. So like my admission to Ohio State ended up getting deferred, but I ended up being able to pursue it in 2021. And then I graduated in 2023, after which I got admitted into Virginia Tech into biomedical engineering again. And I'm currently in the sleepy town of Blacksburg in Virginia. So, my modus operandi or my personal motto is that I seek to integrate engineering and biotechnology to innovate and improve on healthcare. And I will talk more about this in my journey. So, the main why, like for why I pursued biomedical sciences / engineering, has like obviously changed over the years. So, for example, when I was in 10th standard, like it was just something a lot more big picture. So basically, I realized that I liked biology and I had the desire to cure disease, and I specifically wanted to venture into genetic disorders. But when I went into undergrad, I realized that I like translational cancer research a lot more, as well as nanobiotechnology, which I learned through like various electives. I ended up taking, especially in my third and final years. But when I got admitted into my master's and PhD, and as I went through my degrees, I realized that I also like biomaterials and tissue engineering. And it was at this point that I decided that I wanted to get into the pharmaceutical industry, where I could potentially work on like different pharmaceuticals, like for cancer or maybe other diseases. But the common theme I want to present here is that I want to cure disease with innovation. And now, moving on to where it all began. So first year, like it was like full of courses, like we couldn't really take on any projects yet. But as soon as I entered my second year, like in my second semester, I decided to take a study project on genetic diseases. And basically based on what I had liked back in school. And specifically, I had presented a poster on thalassemia and cystic fibrosis at Manipal University in 2018. Then in my third year, when I was finally able to take up lab projects, like in my second semester, I worked under Dr. Menak on this plant called Saliconia bigelovi. It is a plant that can grow in saline conditions, and under those conditions, it can produce like these special chemicals called secondary metabolites, and specifically phenols and flavonoids, which have been found to in like many studies to just like be able to like mitigate disease in higher concentrations. And I had presented a review poster on this in 2019. Then in my final year, like I decided to pivot in more into nanotechnology because that was more my primary interest. And so I ended up working with Dr. Palab for both my first and second semesters. The first semester being making silver nanoparticles, like using casein, which is a protein that is found in milk. And then my second semester, like I decided to pursue my undergraduate thesis on a very different topic, which is on keratin gene expression in E. coli bacteria. So what this basically was supposed to comprise of was I was supposed to take a keratin gene and I was supposed to put it into the structure called a plasmid, which I would then insert into the E. coli bacterium. And through that, the E. coli would be eventually able to express this protein. However, I only got to the stage where I was able to amplify the keratin gene because at that point the pandemic had begun and like lockdown had begun. So but still, like I was still like very happy to see the potential of this keratin gene expression in E. coli, like because it has been found to be like a relatively safer method compared to like generic keratin extraction methods from hair, like which use a lot of chemicals. And so keratin is also like one of the components that can be used to make like scaffolds that are very commonly used to support cells in tissue engineering. So just to slightly pivot, like so some tips that I would like to confer to you guys, especially for those who are currently in your second year and still thinking of what you want to do, especially if you want to pursue grad school. So the first thing, definitely try to think about your interests. Like, so if maybe your interests lie in plant biology, or if you want to do like go into like biomedical engineering, like just try to think about like going off on those terms, and then um, definitely consider like looking into your electives list, like just see what is available every semester and then just contact the professors. I mean, they're always like available to talk to you like about like what the syllabus contains, like and just to see whether this will fit the path you actually want to go about. And then also, if you want to like pursue your master, or if you especially want to terminate with a PhD, it is very, very important to have at least a couple of lab projects or even a thesis under your belt. So the ideal way, like you all could go about this is like you could take a study project. If you like the thing, you could consider doing a lab project. And then you can even consider extending to a second project or a thesis. And with the amount of work you would have accumulated, you could definitely like turn this into a publication, which is very, very, very useful, like for PhD applications, especially, and some master's thesis. So in 2021, like, well, after the height of the pandemic was gone, like, I was finally able to come here to the United States where I began my master's at the Ohio State University. So during my master's, well, as someone who was born and raised in Dubai, like, I was surrounded by certain demographics of people. Like I was definitely not used to like American culture, like the way people talk and all that stuff. And so it did take me a month or two to get used to like everything over there, like especially even like coursework expectations, like on top of exams they expect you to do like assignments and homework, so it is completely different to um, like Bets to Buy. And also you are expected to participate actively in class, like and maybe even like debate a bit with the professor, like which I eventually found out like to be fun. But yeah. So another aspect like of my master's that, you know, I would like to talk about is like selecting a lab. So when I entered my master's, I had interviewed with two professors. Like, so one was doing spinal cord research, and the other one, like, was doing nanotechnology. And both of them seem to have some. I'm sorry. Um, Both of them seem to have like topics relating to the central nervous system, which like I was primarily interested in from the very beginning. Like, however, like I did not like fit in like with the lab culture of the spinal cord lab, and so I ended up joining the lab that was mainly doing nanotechnology, and my thesis was mainly about like DNA containing like self-healing hydrogels. So essentially, like I was gathering mainly preliminary data to make like hydrogels that would heal in the same way that, you know, when we get injured, like our wounds would heal. And the technology that I was trying to use here was called DNA origami. Like when like the DNA hinge, these structures which are called DNA hinges, like they could like open and close if exposed to a certain stimulus. For example, like if you attach like magnetic nanoparticles to the open ends of these hinges, like you can see that if you apply a magnetic field, they can close, and like whatever cracks or cuts might be in the hydrogel would theoretically be able to close. So that was essentially my master's thesis. And so in 2023, I joined my PhD in Virginia Tech, where I'm currently finishing up my second year. And the topic that I'm currently pursuing is a lot more different than what I did for my master's. Like, specifically, I'm researching a very deadly brain cancer called glioblastoma. And so this cancer basically has like a survival time of barely a year, like after diagnosis. And it is so aggressive, and it often like comes back even after like, you remove the tumors and you give treatment that it's essentially guaranteed that people are going to eventually pass. And that is very sad. But more on the scientific side, I'm trying to look at the potential role of a sugar-based coating on the surface of cells called the glycocalyx, because apparently, like dysregulation or like um, basically when there are, um, like different than normal amounts of these glycocalyx components on the surface of cells, like it has been found to be linked to disease. So I'm trying to look at that in terms of glioblastoma. And so I would like to pivot to like giving you all tips on how you can prepare for your master's slash PhD. So, firstly, the tests or courses to take starting your third year in undergrad. Like you can do it a little earlier possible. So, if you're planning on coming here to the US or like going to Canada, then definitely like do your GRE. This is a three-part test, like with verbal components, quantitative components, and essay components, each of which like need their own special dedication to study for, which is why I would recommend trying to study for the GRE at least a year before you take it. So like probably starting the end of your second year, and then maybe towards the beginning of your final year, like or summer between your third and fourth year, like you can go ahead and take the test, because that is exactly like what I did. Then the second thing is definitely English as a second language. So as people like coming from India slash the UAE, like um we will be expected to take these tests as, yeah. So the two tests are like the TOEFL and the IELTS. And also, finally, like this is optional, but it can definitely like boost your resume if you pick the right courses. So, for example, like you want to maybe get into AI in imaging um, for disease. So you can definitely like take like a couple of machine learning courses online. You can even like go ahead and look into the computer science department, see what they're offering, and like take those like courses as well. Like, I mean, if they don't have like too many prerequisites, of course. Like, if they do, then definitely online courses are your best friend, and you can, yeah, go ahead and do those. And also, especially if you want to like, get into biomedical engineering, like a couple of courses I would recommend taking, like especially for like students in biotech are definitely drug design and delivery, like immunotechnology, if that's still being offered. Um, you can even go ahead into drug design and delivery, proteomics. There's a whole plethora of courses that you all can take. Then the second point I'd like to touch upon is definitely finding topics of interest for your master's or PhD. And this is especially important if you want to pursue a thesis, like during your master's. And the first step you can do, like especially before applying to your programs, is to check the lab websites of like the professors. So usually the departments you want to apply to will have their own website, and they will have the section called faculty/staff, which you can check. You can go through the list of professors and see like which professors like have research interests that could potentially fit what you want to do. Then you can go ahead and look at the pages. They will have something called lab websites, which will tell you more in detail about the research. They will also display a list of like perhaps all the publications or maybe recent publications, or even select ones that you can look at that are most important to what they are doing right now. And the second point is especially important, like if you're going for a PhD. So definitely, like once you found your professor of interest, you can contact them, you can ask them like for an interview. Like it's a whole process in itself. So definitely plan out like whose labs you want to get into. And even like sometimes for like some also be important, which is why you need to check like the admission requirements like for the program you want to get into, and just work accordingly. Then the final point is definitely consider like the fit of the program and not just rankings, because see, rankings will definitely tell you a lot like about like the quality of education like from the university as a whole. But like sometimes maybe if you get into a highly ranked program and then you find out that the electives do not fit what you want to do, then that can be an issue. Which is why like I would recommend you look at the coursework syllabus, like which is usually available in something called a handbook on the department website. And it is in those books, in those documents, that you can find out like what electives are being offered, like the specializations that are there. Like I know that like some courses will definitely have specializations. So I would recommend you look into those. And then other than your fees, like definitely like consider the location, like see whether you're okay with like being in a remote like area, like um as long as you like the courses, or if you're like willing to compromise on that a bit to like live in a bigger city where you'd feel like more at home. It really depends on you. And lastly, definitely the potential cost of living, because like tuition in itself is like already like expensive for us international students here in the United States. So, and on top of that, you'll also have to consider the cost of your housing, the cost of groceries. And so all this information is usually available on what is called the Bursar's website. And they will usually give an estimate of the maximum you have to pay like for your housing and stuff. But a lot of the time it is usually cheaper, like especially like if you do end up getting admits, there will be so many WhatsApp groups like for Indians and other international students that you can also inquire into. Like there's so many Facebook pages you can look at to find such groups. And finally, before I move on to the next section, additional tips for grad school applications. So, firstly, definitely make a proper list of what are called reach, target, and safety schools. And this depends on your GPA, GRE, TOEFL scores, etc. So, reach schools essentially means where you have like a less than a 25% chance of getting in with your current profile. That is your GPA and your GRE slash TOEFL. Then there's your target schools where you have around, let's say, around a 50 to 60% chance of getting in. And then there's safety schools where you have anywhere between a 75 and 90% chance of getting in. So you can do that, you can track that like in a spreadsheet. And yeah, then for number two, so I understand like agents can cost like thousands upon thousands of dirhams, but if you can't afford it, definitely consider going through one because definitely understand the process of applying for grad schools. Like they can give you a lot of guidance on what to do, like especially with your whole application package on how to go about a lot of different things because an application in itself can take up to 2 hours to complete at a time. So or maybe even days, like if you spend consider the time taking to gather the materials. But which brings me to point number three, letters of recommendation and statements of purpose. So, l- with your letters of recommendation, these can usually come for like from professors you've taken projects with, um, professors you've taken courses under, and also even like especially if you're doing like PS1 or PS2, you can definitely consider your bosses like at those things. And yeah, so those are some examples. And I would recommend you ask like for them like maybe around, I would say around 5 to 6 months to maybe 3 months before the application deadline. And the reason is professors and like bosses and a lot of professionals, they're very busy people. And especially when you come towards a deadline, which for the US is usually December slash Jan, like they will already have so many letters of recommendation to submit. And like let's say each student like will maybe have like 6 to 7 universities they're applying for, so that's a lot of letters of recommendation they will need to personalize for the student. So which is why I would recommend like asking them like months in advance. Then coming to your statements of purpose, so this is essentially a supplement to your CV slash resume. And essentially this is where you tell the story of your life and not just like replicate what's in your resume or CV. It's basically like you telling the story of how you got where you want to be, what inspired you to get like into the university, like what aspects of like the program are very interesting to you. So again, like I would recommend you get started on those like at least 6 months to 3 months before you like apply like before the deadline. And following up from point number one, definitely keep a spreadsheet to track your applications, like for example your LORs, like SOPs, then the application deadlines, as well as whether you got accepted or rejected. And moving on to point number five, so if you remember what I said in the previous slide, like sometimes like the professors will ask for an interview once you contact them, like to talk about the research, like if they find you interesting enough and they have funding, like especially like if you're looking for funding, definitely like also put like your thoughts on the interviews, your thoughts on the professors on your application spreadsheet, because if you do end up getting multiple admits, it will definitely help you to choose like what is the best fit for you. And moving on to number six, definitely do not forget to ask admissions committees slash your professors of interest of any questions you would have about the lab or research or like even the program itself, because they've definitely been like at the university you've been studying at in the place like the university is located in for a long time. So they will definitely be open to answering questions about those. And if the professor asks you to speak to the current students in their lab, then yes, definitely like ask the students about the same questions as well. Like there are usually a lot of online resources for this, as well as for SOPs, LORs, and like many different things. But what I ultimately want to drive, like in terms of point six, is that you're trying to assess their fit, like, how compatible they are with you, how compatible the program is what you want to do, as much as they are trying to like see how well you fit into the program. So don't be afraid to ask those questions. And now, moving on to the advancements in cancer and tissue engineering. So, the big picture on why we want to advance cancer research is because the global cancer burden is growing by the years, and it is expected to nearly double like from the present time like till 2050 in terms of both like the number of patients with cancer and the number of people dying from cancer. And so this increase in number is expected to like increase the burden on healthcare systems, both in terms of the human labor and the cost of taking care of the patients, as well as the resources needed, which is why, like, biomedical engineers and biomedical researchers in general need to come up with ideas on how this can potentially be reduced. So, the first, like, innovation that has attempted to overcome this is called cancer vaccines. So, if you all know the vaccines like you've received as kids, essentially, like they'll take the virus or whatever infectious agent, which is like called a pathogen, like whatever is causing that disease. They will take a protein from the surface of that cell, which is called an antigen, like, and they will use that to create vaccines to allow your immune system to fight like the disease when it actually comes in. However, cancer vaccines differ from our typical vaccines in terms of that when patient cells are actually being taken, like from the tumor inside the patient, and then those same antigens are being taken from those cells and vaccines are being made that are specific to like the cancer itself. And once the antigens or the formulation with the antigen has been injected inside the patient, like it would essentially like allow like your immune system to actually go to the site of like where the cancer is and actually fight it. And the main why for cancer vaccines is because they are proposed to be a relatively non toxic alternative to current treatments, l- like such as radiotherapy, chemotherapy, and immunotherapy, which are known to have a lot of side effects in patients. And as of 2024, like vaccines that are actually similar to the Pfizer COVID vaccine that pretty much most of us would have taken like back in 2020 or 2021, like they're basically taking like a type of nucleic acid called mRNA, and they're trying to personalize like the vaccines to the patient's cancer genetic makeup, which is why this is indeed an exciting field. And these mRNA vaccines are already under clinical trials. So, moving on to the second point, which is artificial intelligence in cancer. So, basically, data sets of patient data, like such as images of like patient tumors versus healthy tissues, or maybe the presence of genetic mutations in cancers. Um, are used to train like machine learning or AI models, like which will then allow them to characterize like cancers, like according to like maybe their stage, like especially if they see images, if they see um, like genetic makeup, they can definitely figure out what is exactly inside the cells, which will then ultimately lead to like the optimization of treatments for these patient cancers. Then the third innovation that I'd like to touch upon is ultrasound waves of cancer therapy. Fun fact, one of the professors in my department is actually like big in this like research field. And so it basically works on the basis that, in ultrasound, like frequencies of about 20 kilohertz are generally used, like to like target tissue, like either like for imaging or for treatment. But research has found that certain frequencies of ultrasound have been found to be used for cancer therapy. And how this works is that when these ultrasound waves are focused on the tumors, like the fluid surrounding the cells, which is called like interstitial fluid, like it will form bubbles, and those bubbles will burst at such a high intensity that it will literally destroy the tissue in the same way an explosive can destroy a wall. And so if you can see like above the above image, um, these are like the little bubbles that are called like cavitation bubbles that are like targeting the tissue. So yeah, especially in figure B. And research has all the same research has also found out that this ultrasound therapy has also like positively stimulated the immune system, which then clears like the resulting debris. So the last point I'd like to touch upon in terms of cancer innovations is that um, it essentially uses the patient's own like T cells, which are a type of white blood cell, to fight cancer. So, firstly, the patient's blood is taken, okay, as you can see over here, and then the T cells, which are known to directly like act upon cells, um, they're basically reprogrammed. After which, like, they are exposed to cancer cells where they learn to target them. And once those certain cells which can target the cancer cells have been found out, they can be expanded, which means they're grown into the millions upon millions, like of like identical such cells, which are then injected back into the patient and will therefore allow them to fight the tumor. And now I would like to move on to tissue engineering innovations. So, what is tissue engineering? So, essentially, it combines like both like the biomedical sciences as well as like different concepts of engineering, like to be able to create technologies that can restore or maintain or even like improve like tissues that have been damaged through either like disease or just injury. And so, some of these technologies include like iPSC. These are induced pluripotent stem cells, which can basically be turned into nearly every cell type in the body, like through exposure through different chemicals. Then there's reprogrammed cells, which like take cells from, for example, I know the most common example is the skin. So they take cells from the skin, they remove like all the genetic coding that causes skin cells to be skin cells, and then they reintroduce like a different kind of genetic programming that turns the cells into the desired cell type, like such as liver tissue. Then there's bioreactors, which can grow millions upon millions of cells. Then there's scaffolds, which like provide like support, like specific support to different cell types. And then finally, there's 3D bioprinting, which combines like different biomaterials as well as cells to make different like tissue or even organ constructs. And there's decellularized organs which just have the scaffold part of the organ, which is called the extracellular matrix present. And these organs can then be repopulated with the patient's own cells. So now for the why in tissue engineering. So traditionally in biomedical research, like a lot of like animal models such as mice, dogs, like and even monkeys have been used to research like different diseases. However, like there is only like a certain like and that too it's usually a very small number of animals that is generally the limit kept by ethics committees. And ideally, like ethics committees would rather you use a non-animal like model to do your research, which is where tissue engineering comes in to help like make models that simulate like human physiology as much as possible. Then the second point I'd like to touch upon is the lack of viable donor organs because like usually like especially like if you watch medical dramas like you'll find that um there are so many episodes in which the patient is kept on a transplant list and they're waiting and waiting and waiting. So we need to like find ways to like replace those like the need for donor organs and therefore get patients off the waiting list like as fast as possible. And so one of the technologies that has recently been found to as an innovation is called scaffold free tissue engineering. So if you remember the scaffolds I described earlier, so these are usually made of like many different kinds of biomaterials, like from natural which are found within the body to maybe some like slightly synthetic such as polyethylene glycol. But the problem with some of these materials is that like when they are introduced into a patient's body, like they will cause what is called an adverse immune reaction, or the immune system will essentially attack like the cells in those scaffolds. So in order to find a way to you know just bypass this, researchers have found something called scaffold free tissue engineering, which relies on the ability of cells to produce their own scaffolds to form a niche called a microenvironment. And so like the cells like they will secrete their own materials, they will form their own supports essentially, like to form what is called an extracellular matrix. And if you can see at the bottom right, so this is not exactly a true example, but these are actually the cells that I'm studying. They're called glioma stem cells. And so the cells they can grow amongst themselves and they generally attach to each other. So essentially they become balls of cells called spheroids, and these are usually stuck to each other using their own extracellular matrix. Although this is um yeah, this is an example of scaffold free tissue engineering. Then the next innovation in tissue engineering is called smart biomaterials for tissue formation. So um, there have been like different biomaterials that have been brought about like in recent years that they can respond to changes in pH, like they can respond to changes in the mechanical environment, which is like especially important in certain organs such as bone, and they can even interact like with cells to guide and enhance tissue penetration. And so what happens is that when these biomaterials are exposed to certain environments, they can like sense the changes, and this then triggers the release of like many different materials. So it can be maybe materials to repair the tissue, it can be materials to just like allow cells like more cells to grow there, and so on and so forth. And finally, CRISPR slash Cas9. So well, we all know that Jennifer Doudna, like won the Nobel Prize for CRISPR slash Cas9. I believe it was back in 2020 or 2023. Yeah, I think it was 2020. But I digress. And CRISPR allows us to directly reprogram cells to generate tissue. And the basis on how this works is similar to what are called bacterial immune systems, where they actually take viral DNA and they integrate it into their own DNA structure, so that when the same viral DNA comes in, like they can fight off that whatever the viral infection is. And so, using the same concept, RNA strands called guide RNA strands can be inserted into the DNA of different cell types, like usually stem cells, because they have not been developed into what's that? Their final form, essentially through differentiation. So this can this RNA can either knock down or deactivate the genes, or it can be used to activate or just allow the gene to be expressed more. And so stem cells have been used to generate in vitro models to study different diseases, especially like and I do know of a few examples where they've actually been used to make different cells in the central nervous system to allow them to study diseases like Alzheimer's and multiple sclerosis. And now, finally, the current biotech trends and market. So, I will just briefly touch upon this. So, there's AI in biotech, where it's increasingly being used for diagnosis and also potentially for drug discovery and development. Then there's gene therapies, for example, in the form of vaccines, such as the cancer vaccines, like I mentioned earlier. Then there's precision medicine, which can then be tailored to a patient by taking, the cells of the disease that they have and just making decisions on the exact concentrations of drugs or whatever to give to the patient. And finally, sustainability in terms of the materials being used to make the pharmaceuticals as well as the packaging. And with that, I conclude my presentation and thank you for listening to my talk. And I'm open to any questions, and my contact information is below.
That was a very insightful talk, Samantha. Thanks for coming. And anyone has any questions, they can raise their hands. I had a question. And at this stage, a lot of us are confused to choose either thesis or PS because we don't know whether we want to do masters. Like we are not we didn't think of it. A lot of us want to do jobs for a few years and then shift to masters. So what do you prefer? Like what should we do, like thesis or PS?
So I guess if you want to go into a job, I've heard that especially from my batch, a lot of people have decided to go for PS instead of thesis. If especially if they want to make connections over here in Dubai or maybe back in India. So I would recommend PS more. And honestly speaking, this is something I forgot to touch upon in the presentation. Work experience is also very, very useful, for getting into masters, and a lot of universities will see that as a positive. So
Right, right.
yes.
Mm. Okay. Anyone has any question?
Hi, Samantha. That was a really informative session. Thank you so much, firstly. I had a question about, the recent trends in sustainability and how we could move forward if we want to, pursue a career in sustainability and that sector.
Mhm. So if you're looking like to maybe pursue research in sustainability, like if you're looking to maybe find ways to make like greener pharmaceuticals, like I guess a couple of courses that I would recommend you take like are would be along the lines of drug design and discovery and definitely biomaterials, like, and not just like medical biomaterials, like probably just general biomaterials in like general. um it would be preferable if the course like potentially focused like or at least had like section that focused on environmental like biomaterials so that you know you could have an idea. Like I'm not sure if this entirely answered your question, but I hope it did.
Yes, it did. Thank you so much.
Also, how is the job market like in biotech in US? I guess? Comparing Dubai, like, is Dubai a good place to pursue your career in biotech, or there are better countries? Of course, there are better countries, but then the people who just want to like pursue their career in Dubai, is it a better place to consider?
I mean, definitely not as much as the Western world, but there are definitely like an increasing number of companies in Dubai that I've heard of. Like, in fact, I've heard of like multiple alumni from BITS itself. I think maybe my batch or close to that that are currently working. I mean, I definitely do not know what the exact job market is like, but you can definitely go ahead and try looking for those people. Like, and yeah.
Oh, right, right.
Asvi, I guess if we don't have any more questions, we can end the call.
Yeah.
All right. Thank you, Samantha, for the amazing talk today. It was nice hearing from you.
You're welcome, and I actually thank you and the Google Developers Student Club like for inviting me to like present like my experience to you guys.
Absolutely.
Thank you.
All
Also,
right.
Thank
Samantha,
you.
yeah, could you share your PPT with us if you don't mind? Like, it has a lot of resources. Yeah.
So what I'll do is like I'll send the PPT to Anushka. I will need to edit it a bit before I can send it off, but like I will let Anushka know once I
Okay.
send
Yes.
it to her.
Okay.
Yeah.
Yep.
All right. Thank you. See
Thank
you.
you so much. Thank you.
Thank you. Bye
Bye.
bye.
Bye-bye.
Asfia.